Hello everyone, welcome to the eLab Science Flow Cytometry Practical Guide. Today's learning topic is, how to efficiently design a flow cytometry experiment. The first step in experimental design is to clearly define your objectives. To do this, you need to address the following questions. What species is the study subject? Is it human, mouse, or another organism? What markers need to be detected? Are they surface markers, intracellular signaling molecules, or nuclear markers? What is the expression level of the target markers in the sample, and what is the relationship between these markers? For example, when analyzing immune cells in human peripheral blood, the study subject is human peripheral blood, so antibodies need to choose anti-human. Markers to be detected may include CD45, CD3, CD4, CD8, CD25, CD127 FOXP3, IFN gamma, CD19, CD16, and CD56. The selection of markers should align with the study objectives. We take the detection of T, B, and K cells as an example. Markers such as CD45, CD3, CD4, CD8, CD19, CD16, and CD56 can be chosen. These are all surface markers and do not require fixation and permeabilization reagents. However, if markers such as FOXP3 or IFN gamma are included, appropriate fixation and permeabilization reagents will be necessary. Finally, it's essential to determine the expression levels of these markers and their relationships. Step 2. Selecting fluorochromes. The choice of fluorochromes should consider the configuration of the flow cytometer and the principles of panel design. Flow cytometers on the market vary significantly, with configurations such as single laser, 4-color systems, dual laser, 6-color systems, dual laser, 8-color systems, or even 3-laser, 13-color systems, and full-spectrum cytometers. Begin by identifying the available fluorochromes for your instrument and then determine the optimal fluorochrome combination based on panel design principles. Using the Agilent NovoCyte 3000 flow cytometer as an example, for detecting CD45, CD3, CD4, CD8, CD19, CD16, and CD56, you can pair fluorochromes based on the configuration of the flow cytometer as follows. Step 3. Setting up controls. Controls in flow cytometry are critical for accurate interpretation and can be categorized into 5 main types: blank control, single stain control, isotype control, fluorescence -1 control, biological control. Final steps. After designing the experiment, incubate the flow cytometry antibodies, optimize the instrument parameters such as channels, thresholds, voltages, flow rates, and proceed with data analysis. Efficient flow cytometry experiment design requires clearly defined research objectives, careful selection of antibodies and fluorochromes, appropriate control setup, and optimized instrument parameters. This concludes the content of this video. Stay tuned with eLabScience to learn more about flow cytometry knowledge.